In this video, we'll break down the key bioinformatics file formats for sequences and alignments. If you're learning bioinformatics or working with DNA, RNA, or protein sequences, knowing these formats is essential. We'll cover both sequence formats, like FASTA and GenBank, and alignment formats, like CLUSTAL and SAM, in a way that's easy to remember. Let's start with sequence formats. These are files that store raw DNA, RNA, or protein sequences. FASTA, probably the most common format. Each sequence starts with a bigger-than header, followed by the sequence itself. It's simple and widely used for bioinformatics tools. FASTQ, similar to FASTA, but includes quality scores from sequencing. Each read has 4 lines, a header starting with the sequence, a plus line, and the quality scores. GenBank, this is a rich annotation format, storing sequences along with features like genes, coding regions, and more. It's widely used in NCBI databases. EMBL, another annotated format, mainly used in European databases. It's similar to GenBank, but with slightly different headers. GFF or GTF, these are feature formats, storing information about gene locations and other genomic features. They are tab-delimited files that indicate positions, gene IDs, and types. PIR, a protein sequence format, sometimes used in older bioinformatics pipelines. In short, sequence formats are about storing the actual sequences and any useful annotations for analysis. Next, we have alignment formats. These store sequences that have been aligned to find similarities, conserved regions, or evolutionary relationships. CLUSTAL, a classic format showing multiple sequences aligned in blocks, with symbols indicating conserved regions. PHYLIP, column-based alignment format often used for phylogenetic tree construction. Stockholm supports alignments with annotations, such as secondary structures for proteins or RNA. MEF, multiple alignment format, commonly used in genomics for whole genomes. SAM or BEM, SAM is a text-based sequence alignment map for reads mapped to a reference genome, and BEM is the compressed binary version. Essential for next-generation sequencing workflows. VCF, variant call format, used for storing differences or mutations detected in alignments. These formats are critical for analysis, whether you're comparing sequences, building phylogenies, or analyzing genomic variants. To wrap up, knowing these file formats is essential for any bioinformatician. Sequence formats like FASTA and GenBank store raw data, while alignment formats like CLUSTAL and SAM store relationships between sequences. Using the right format ensures smooth analysis and accurate results.